I really like to bring new solution, new equipment to the researcher. And they always need new development to find new protein, to find new assay. And this is something I really like, to play with the protein in the fundamental research and develop a test which give you the clinical trial at the end and maybe change the life of the patient. This is my reason to come to work. So I'm Alexandre Lucas. I'm the founder and manager of WeMet. This is a facility focused on protein analysis and biochemistry. I'm working at INSERM, the French National Institute of Research, in the N2MC here in Toulouse, which is an institute focused on metabolic cardiovascular diseases. So there is a 15 team who works on different topics around that and try to find new candidates for treatment, and new signaling pathway, and many things like that. First thing is work with a little amount of sample, which is quite impossible with the classical Western. Another example, we could work with high molecular protein, with low molecular protein, which is quite difficult with classical Western. There is a lot of issue with the traditional Western. The ethics issue, some manipulation of the image. So we need to move on a simple Western because it gives you robust result. And this is the final goal, to get some impact on research. The first time it's quite difficult. You're thinking, oh, it's a completely new way of thinking about the Western blot. Oh, it seems a strange plate to fill, but after that, they will not come back to the classical Western blot. And yes, they like everything, the robustness, the precision, and they also like to have the ability to test and make a good assay on Western, to test saturation, linearity in 3 hours. In classical Western blot, you couldn't do that. It takes you a week. Multiplex detection we used is mainly focused on this ability to go to the near infrared and the infrared possibility. To us, the best advantage of using this multiplex technology, it's really about the cell signaling when you want to look about a phosphoprotein regarding the total protein. So, for example, phospho-ERK and ERK, the classical one. It's really amazing because you could use the chemiluminescence pathway, the signal for the phosphorylated form, and the near infrared for the total form in the same capillary. It could give you really an answer of percentage of activation of your protein, and this is something we couldn't do. We choose the Leo for the high throughput capacities because with this large amount of sample, 2,000 patients, it's really the first objective for us to assess 100 patients in 3 hours. It's amazing because you don't have the variability of the equipment. This is why the first thing for this type of study, the Leo is the best. And we also look at that with the result, because we get 5 or 6% of CV's variation, which is quite amazing, honestly. So we make a project on cell and gene therapy. So we used a standard curve with a recombinant protein, and we analyzed 100 samples to estimate with accuracy the quantity of the protein, like an ELISA, but an ELISA on tissue and denaturating protein, which is completely amazing. The most important things to me, if we move on to the clinics at the end, if we want really have a huge impact of future clinical research, clinical trial, we need to have something to give absolute quantification. With the Leo, you could do that. And with the throughput of the Leo, you could assess finally 70 samples with a duplicate standard curve, and it gives you the precise amount of your protein inside the sample. This is the future for the clinical research, because clinicians need this quantification to make the diagnostic, to move on therapies. We have a lot of projects coming on the Leo, but if I could choose one, this is one done with the Collège de France in Paris, We will try to assess the absolute quantification of one protein inject direct in the nerves in gene therapy finally. And it will be amazing because we could follow finally the absolute quantification of the protein during the different part of the nerves. And this is something really amazing. We could do that with the Leo because we will have 500, 600 part of the nerves to really be capable to say, yes, if we put this amount of protein, the protein go until this part. And the question is, it will be useful or not for the patient at the end. This is the next project I really like because we could really validate the therapy with the Leo and with this huge amount of sample, but I think the Leo is the future about this type of translation to the clinics.